Hello, good evening, class. We want to start our last lecture on basic biochemistry practicals. So, we'll be looking at the Barrett test. So, this is our expectation. For the Barrett test, a blue color means what? Absence of proteins. Whereas a deep purple color means what? Presence of what? Proteins. So that's a positive test. Color reactions are due to a reaction between one or more constituent radicals, or group of radicals, or group of complex protein molecules in the chemical reagent used in any given test. Most of the color reactions are not used to determine the protein in urine, because urine itself is colored, and secondly, its inorganic constituents is colored, and thirdly, its inorganic constituents, example sulfates, interfere with the color reactions. To detect the presence of proteins in the given solution, it is a general Barrett test is a general test to determine the presence of proteins in a given solution. So if you want to find out whether there is protein in a given solution, we use the Barrett test, and it detects the presence of peptide linkage in polypeptide or proteins. Compounds containing two or more peptide bonds give a characteristic purple color when treated with dilute copper sulfate in an alkaline solution. The name of the test comes from the compound Barrett, which is the simplest compound that gives a typical positive reaction. The structure of Barrett shows clearly that it is formed from two molecules of urea when heated at 180 degrees. The exact reason of the color is not known. According to one common view, the color is due to the coordination complex of the compound of the copper atom with 4 nitrogen atoms of a protein. So these the reagents that it requires. The procedure is spelled out here, so go through it in your slides. Interpretation of test results. A purplish violet or pinkish violet color indicates the presence of protein in the test tube. If the color becomes blue, it indicates negative test. So you can see this is the results. Blue color is negative, deep purple color is positive. Peptone gives pink color and may require the addition of a larger amount of copper sulfate solution than usual. Gelatin may give a very faint color, and therefore control is required to avoid confusion, because gelatin will give a faint color. Precautions. Excess of copper sulfate should be avoided. That's the reagent we require for this procedure or for this test. Too much of copper sulfate reagent should be avoided. Otherwise, the blue color of cupric hydroxide, because copper sulfate itself is blue color, so its constituent cupric hydroxide will mark the violet color. Magnesium sulfate and ammonium sulfate interfere with this test. Therefore, the test should not be carried out with solutions containing these salts. The test may also be given by substances containing a peptide bond, whether it is in protein or in non-protein compound, such as Barrett and oxamide. So the test may also be given by substances containing a peptide bond, whether it is in protein or non-protein compound. So So if it's even in a non-protein compound and there's a peptide bond, the test results will show. Reactions of reducing and non-reducing sugars. Reducing and non-reducing sugar, we have oxidation, that is loss of electrons, reduction, gain of electrons. So reducing sugars, these are sugars that can be oxidized by mild oxidizing agents, and the oxidizing agent is reduced in the reaction. You remember your redox reactions I taught in first semester. So a non-reducing sugar, these are sugars that are not oxidized by mild oxidizing agents. Sugars exist in solution as an equilibrium mixture of open chain and closed ring structures. All monosaccharides have an open and closed form structure, but oligosaccharides have only closed structure. So sugars, open chain, closed ring. We did this one earlier on when we were tackling carbohydrates. So you can see all monosaccharides have an open and closed chain structure. So this is a monosaccharide, this is the open chain, this is glucose, this is the open chain structure, and this is the closed chain structure. The active part in sugars is aldehyde or ketone group. So every sugar has an aldehyde or a ketone group. So let's see what an aldehyde is. So for instance, if you have CH3, C double bond O, and C double bond O is what you call the carbonyl carbon. Hey, it's what you call the this carbon is the carbonyl carbon. This carbon is the carbonyl carbon. So let's plot this. So if you have another CH3 here, then this one becomes what? A ketone. This is a ketone. But if you have CH3, C carbonyl carbon here, and you have what? Only H here, then this whole structure becomes an what? Aldehyde. So the structure of a ketone is represented as this: R for alkyl group, double bond O, R. Then aldehyde has what? R, C double bond O, then H. So instead of R here, there is what? H here. So this is an aldehyde, and this is a ketone. So ketone, this is the carbonyl carbon. This carbon is the carbonyl carbon. This carbon is also the carbonyl carbon. But here we have H instead of an alkyl group. Lactose is reducing sugar. One of the carbonyl groups are free, as shown. So if there is if a compound is a reducing sugar, it means that one of the carbonyl carbon, that's carbon 1 here, should be free. So for lactose, this one is free. Carbon 1 is free. But sucrose is non-reducing sugar because carbon 1 is not free. No, sucrose is made up of glucose and what? Fructose. But you realize that carbon 1 of glucose is occupied, carbon 1 of fructose is also occupied. So it's a non-reducing sugar. It's occupied in a bond, it's not free. Like the lactose, which this one is free, carbon 1 is free. For the sucrose, carbon 1 in glucose and carbon 1 in fructose are not free. Starch and polysaccharides are non-reducing sugar because they don't have carbon 1 being free. Common oxidizing agents used to test for the presence of a reducing sugar are, there should be what? A free anomeric carbon, that's carbon 1. So if carbon 1 is free, then we can use Benedict solution, Furling solution, and pricklic acid solution to find out whether it's a reducing sugar or not. So common oxidizing agents used to test for presence of a reducing sugar. That's reducing sugar means that there should be what? Free anomeric carbon. Benedict solution, Furling solution, and pricklic acid solution. Benedict solution or Benedict reagent contains cupric ions which in an alkaline environment oxidize the aldehyde group to a carboxylic acid. So the aldehyde group is converted. This aldehyde group is converted to what? Carboxylic acid. That's LCO. Then we add what? O to it for making OH. This is what? Carboxylic acid. This is carboxylic acid. Cupric ions are reduced to cuprous oxide, which forms a red precipitate. So this is the carboxylic. This aldehyde, in the presence of two molecules of cupric ions plus 4OH, are converted to alkanoic acid or carboxylic acid. These are the reagents. This is the procedure. This is the color change. So at the end of the reducing sugar test, like Furling's, Benedict's, and the rest, we are supposed to have what? Blue coloration means none, there is no reducing sugar. Green to yellow coloration means there is trace or very small amount of reducing sugar. Moderate means there is what? Moderate amount of reducing sugar. Then brick red precipitate means a lot of reducing sugar. Substantial amount of reducing sugar is present. You can also see this is the brick red. That means substantial amount of reducing sugar is present. But the blue means there's no reducing sugar. Let's look at the Xanthoprotect test. Xanthoprotect test is for the detection of aromatic amino acids, tyrosine and tryptophan. So if you are looking at the Xanthoprotect test, we want to just determine the presence of what? Tryptophan or tyrosine. So if tryptophan has, this is the aromatic group, the benzene ring. This is the benzene ring. Another benzene ring is here. So tyrosine has a benzene ring. Tryptophan also has a benzene ring. So it will respond to Xanthoprotect test. On addition of concentrated nitric acid, the white precipitate is formed due to denaturation of proteins. The benzene ring, that's the phenyl group present in tyrosine and tryptophan, undergoes nitration on heating with nitric acid. The nitrophenyl group imparts yellow color due to the solution. So a phenyl group, this is a benzene ring, or also called phenyl group. Phenyl group. This ring is phenyl group or benzene ring. On addition of ammonia, the nitrophenyl group ionizes, imparting orange color to the solution. Nitration of phenylalanine does not take part under these conditions. Therefore, it does not respond to this test, although it also contains benzene ring. So nitration of phenylalanine is not part. It doesn't take part under this condition. Therefore, it's not okay. So these are the reagents. So you have to go through the procedure, go through the reagents also. We may ask you questions. I may ask you questions on them. Then the interpretation of results. So appearance of deep yellow or orange color will indicate the presence of aromatic amino acids, tyrosine and tryptophan in the protein. Gelatin, which contains no tryptophan and very little tyrosine, gives only a faint color. So let's see. For the Xanthoprotect test, the positive test is what? This is the negative test is here. Positive test. So this is a positive test. This is a negative test. For the positive test, we have presence of aromatic amino acids, tyrosine and tryptophan. Presence of the dark yellow or orange color. But with the negative, we have absence of dark yellow or orange color because there is absence of aromatic amino acids, tyrosine and tryptophan. Let me draw an aromatic amino acid and you see. So we have this. This is the benzene ring. Benzene ring. So you can have it here, like this, like this. Or we can also draw it like this. I can also draw it like this and put a circle inside. It's also a benzene ring. So we call it phenyl. Phenyl group or benzene is the same. Either benzene or phenyl group is the same. Phenyl. It is also called aromatic group. Also called aromatic group. So the Xanthoprotect test, look for the presence of what? Phenyl group or aromatic group or benzene ring. Benzene ring in what? A mixture. So you can see Xanthoprotect test is for the detection of aromatic amino acids, tyrosine and tryptophan. So you can see this is the structure I drew. Is the same here. So we look for this ring in an amino acid mixture. Okay, so kindly go through the slides. Well, so that means that you have 3 slides. You have 3 different subjects for basic biochemistry practical. That is the glucose determination, GC-PD test, and color reactions of carbohydrates, color reactions of amino acids, and also we have reducing sugars. So go through them. Make sure you familiarize yourself with the slides also as you take the quiz. So hopefully Friday we'll start the quiz on biochemistry, biological chemistry, and basic biochemistry practicals. So we'll do it on Friday, if possible, Saturday and Monday. Then we'll continue. Tuesday we'll do a quiz, Wednesday we'll do a quiz, Thursday we'll do a quiz. Then it will translate to the next 2 weeks, Friday, where we start the immunology quizzes. Please learn very hard because the exams is going to be very difficult. If you want to fail, you know the things are many. So if you try to fail, I will fail you. If you fail, I will fail you. Imagine you going to read these plenty things all over again. Look at the psychological problem it will give you. So try and just kill the animal. Try and overcome this burden. Try, because the slides are very educative. Thank you very much.